genetic counselor with Stanford Children's, who's going to share a little bit more and be available to us to answer any questions about it. Lauren?
Thank you. And first of all, of course, Deborah, thank you so much for sharing your story. As a genetics provider, I'm always just so humbled and touched to hear your story. So thank you. So hi, everyone. My name is Lauren Skank. I'm a genetic counselor, as Carrie mentioned, with the Pediatric Neurogenomics program at Stanford. And my slides are going a little bit crazy here. Next. Okay. Oh.
I am so sorry about this. I don't know what is exactly happening. It seems to want to go on its own. I apologize so much. Let me try.
That's all right. Let's try one more time.
No, it's okay. Well, I apologize to everybody. I'm going to pause it for a second. So Lauren, if you don't mind sharing a little bit more about yourself while I take this offline and see if I can repair it.
Oh, sure. Thank you. All right. Well, and I can also just go ahead and get started and talk through the first couple of points. So today I'll be talking about GM1 genetics and your family planning options. And so first of all, I just wanted to go over what kinds of topics we'll be talking about today. And so the first thing I wanted to talk about was sort of foundations of genetics, or sort of genetics 101. Next, I'll talk about diagnosing GM1, as well as the inheritance of GM1. And then we'll really get into the bulk of the presentation, which will be about family planning options. And we'll talk about genetic testing before, during, and after a pregnancy.
Okay, let's try this again. Hold on one second.
Sure.
I hope this works. No, it's, sorry, there seems to be automatic transitions in yours. So I'm going to just do it this way, if that's okay with everybody. Can you see? Okay. No. I'm just going to do it. I apologize to everybody for the technical issues, but I'm just going to use it this way for now, if that's okay, Lauren.
Sure, that's just fine.
Okay,
all right, next slide. No problem. So I'm going to really briefly touch on the sort of the foundations of genetics. I know you've had a lot of genetics or science talks already in this conference, so I'll move through this pretty quickly. So I think of genes as the instruction manual for our bodies, and it's made up of DNA sequence A's, T's, G's, and C's. And our DNA or our genes are then packaged into chromosomes. And you can see a picture of chromosomes on the slide on the right-hand side. And our chromosomes come in pairs. In each pair, one comes from mom, one comes from dad. And that also means that our genes come in pairs. One from mom, one from dad. Next. So briefly, what is a genetic mutation? It's a spelling error in a gene that causes the gene to not work properly. And another term for that is pathogenic variant. And I mention that because you may see it on a genetic test result. Next slide. So now talking just a little bit more specific to GM1 gangliosidosis, I'm sure many of you, or maybe all of you, are already aware it's an inherited lysosomal storage disorder, affects the nerve cells in the brain and the spinal cord. It's caused by mutations in the GLB1 gene, and the GLB1 gene provides instructions for making an enzyme that's called beta-galactosidase, which again plays an important role in the brain. When beta-galactosidase enzyme doesn't work properly, GM1 gangliosides accumulate, again, especially in the brain. And you can see in the picture on the bottom, so here the GLB1 gene, it becomes transcribed and translated and becomes a protein that has a function in the body, and that protein in this case again, is the beta-galactosidase enzyme. Next slide. So how is GM1 diagnosed? And there are two different ways it can be diagnosed. One is through looking directly at the enzyme activity, and the other would be looking for pathogenic variants or mutations in the GLB1 gene. So enzyme testing can be used through a blood test, and it's looking at how much of the enzyme is produced. The genetic testing can be done through either blood or a saliva test, and it's looking for spelling errors in the genetic sequence. And there are a number of different ways that genetic testing can be done, whether that's specific for one gene, just for specifically GLB1, or if it's looking more broadly at multiple genes at one time, which could be with either a multi-gene panel that looks at many genes at once, say in the range of 50 or maybe multiple hundreds of genes. An example of that would be a leukodystrophy panel. Or another broad test would be exome sequencing, looking at potentially 20,000 genes, all of the protein coding genes in the body all at once, looking specifically with relation to your child's symptoms. For family planning purposes, genetic testing is important. First, we have to identify the mutation in your child with GM1 before being able to test other family members or a pregnancy. Next slide. So briefly, I want to review some genetic inheritance patterns. So on the right-hand side, or the left-hand side, excuse me, you can see autosomal dominant inheritance. And again, we have 2 copies of every gene in our body, one from mom and one from dad. Dominant conditions occur when just one copy of a non-working gene caused the condition. On the other hand, autosomal recessive inheritance is when the condition is caused by 2 non-working copies of the gene, one from mom and one from dad. And GM1 falls in the recessive category. Next slide. Thank you. So GM1, again, autosomal recessive, and that means that there's a 25% chance or a 1 in 4 chance to have a child with GM1 when the 2 parents are carriers. Next slide. So you may be wondering, what does all of this mean for our family in particular, and what options do we have if we're considering having more children? Next slide. So first of all, I just want to say that each family is totally different in terms of what options they might consider, and there's no right or wrong option. It just depends on your own family's values and beliefs. So I'll review the options on this slide first and then go into more detail on the following slides. So some of your family planning options would be unassisted conception with no prenatal genetic testing. Another option would be genetic testing before a pregnancy using in vitro fertilization or IVF and pre-implantation genetic diagnosis or PGD. Another option is testing during a pregnancy using chorionic villus sampling or CBS or amniocentesis. Or some families consider using a sperm or an egg donor or adoption. Next. So I'll go into each one in a little bit more detail. So unassisted conception without any genetic testing. Again, there would be that 25% or 1 in 4 chance of having a baby with GM1, and that's true for every pregnancy. One of the important things to know is that genetic testing is still available at birth, so that we can make sure that your baby would receive all the proper support and medical services right away. So some of the pros with this is that it avoids medical procedures before or during the pregnancy. And a con would be that it does not allow for knowledge of GM1 before or during a pregnancy. Next slide. Another option, again, this is genetic testing that can be done prior to the conception of a pregnancy, is IVF, which stands for in vitro fertilization, along with PGD. And this involves, you may have heard of IVF before as well, in relation more to couples with fertility issues. So it can be used for people who don't have fertility issues as well, in order to allow for genetic testing of embryos prior to conception. So how this is done is the first step is ovarian hyperstimulation in the woman, and that helps, uses injections and medications to help the woman produce more than one egg at a time. Then a procedure is used for egg retrieval, and a sperm sample is obtained as well. And then the sperm sample and the eggs are combined in a petri dish to allow for the embryos to form. And then we move down here in the bottom picture, you can see the PGD steps. So the embryo grows, the cells divide and multiply from a 1-cell stage, 1 to 2 to 4 to 8. And around the 8-cell stage, one or a few cells can be removed from that embryo to test for GLB1 mutations. Then an embryo can be chosen to be transferred back into a woman's body, and then hopefully a pregnancy occurs. So some of the pros with this option is that it allows for selection of embryos without GLB1 mutations. It allows for diagnosis prior to conception. And cons are that it is expensive, and for some families it can be prohibitively expensive. And there are significant medications, injections that are used for IVF to stimulate the woman's egg production, which can be stressful on a woman's body as well. Next slide. So now I'm going to talk a little bit about prenatal diagnosis options, and the first one being chorionic villus sampling, or CVS. This is a test that can be done looking for GM1 in a pregnancy at 10 to 13 weeks. And it involves genetic testing of the placenta, and specifically it's the outermost part of the placenta, which is called the chorionic villi, thus the name chorionic villus sampling. The procedure can be done in one of two methods, depending on where the placenta is located in a woman's body. So if the placenta is located closer to a woman's backside, then a transcervical procedure is used, and they insert forceps, pretty similar to a pap smear, and then use a small syringe to obtain a sample or almost a biopsy of the placental tissue. And they have the ultrasound on the entire time. With the transabdominal option, that's done when the placenta is located closer to a woman's front side, and they use a needle to obtain, again, a small sample of the placenta. With this procedure, there's a risk for miscarriage that's estimated at less than or equal to 1 in 100. You do see different numbers for the risk of miscarriage in different studies, with some studies showing much somewhat lower risk than that in these newer studies. But it depends on the facility that the procedure is done at, what numbers they use. Next slide. Another option for prenatal diagnosis is using an amniocentesis. And this can be used to test for GM1 in a pregnancy at 14 to 20 weeks. And it involves genetic testing of the amniotic fluid, which is essentially the fetal urine that's passing through the mouth of the fetus, through the lungs, and then passing back out. And it contains fetal skin cells, which have DNA. With this procedure, again, they use ultrasound on the abdomen and a needle to go through the abdomen into the uterus to obtain a sample of the fluid. With the amniocentesis, risk for miscarriage is approximately less than or equal to 1 in 500. Next slide. So some of the pros of using a prenatal diagnostic option is that it allows for genetic diagnosis of GM1 during a pregnancy. It is less expensive than IVF and PGD. It can, a couple can still become pregnant without using reproductive technologies as well. Some cons is that it does not allow for testing even prior to conception, and there is a small risk of miscarriage associated with either of these procedures. Next slide. Another option is to use a sperm donor who does not have a GLB1 mutation. And a sperm sample is obtained, and then they use intrauterine insemination, called IUI, or other insemination techniques to insert the sperm directly into a woman's body. Carrier screening can certainly be done in sperm donors, and that can involve either testing just for the GLB1 gene really specifically, or sometimes using a carrier screening panel that looks at many genes at once that can be associated with recessive conditions. So some pros of this is that it allows for selection of sperm without GLB1 mutations. It avoids IVF and it's less expensive. And some cons is that, one thing to mention, this is a con for some people, but certainly not for everyone, is that the baby would not be biologically related to the father. Another note on this is that you can also use an egg donor, but it's more expensive and requires more medical procedures. And for that reason, it's a bit less common. Next slide. So one other option, of course, is adoption. Pros is that it avoids medical procedures and the associated expenses. The cons, again, for some people, is that the baby would not be biologically related to either parent. And of course, there are financial and emotional costs associated sometimes with the adoption process itself. Next slide. So there are many options available to you, and each family may feel very differently about which ones they would consider. And I think prenatal genetic counseling to really help understand those options and weigh the benefits and limitations is important and can be very helpful for families, and prenatal genetic counselors can also help facilitate some of these options as well. So, next slide. So, thank you so much for your time and for listening today.
Thank you so much, Lauren. And I am so sorry again for all the little chaos.
No problem.
I'm going to see if it's going to work now. Oh, sure. It works on your final slide. So we did receive a couple of questions through the chat. If anybody else has other questions, please send them through. We'll share them with Lauren at the end so she can answer them. And I also just want to reiterate, for those of us that have been working in rare disease advocacy, especially parent advocates, like, I have done for like the past 10, 12 years, genetic counselors are